So we enter back into the alimentary canal of the small intestine. We took a little side route for some accessory organs, including the liver and the pancreas, but now we're back in the small intestines where the majority of all the work associated with digestion is completed. Up until now, the primary digestion that we've been completing has been digestion that's broken big molecules into medium-sized molecules. The remaining amounts of digestion is going to be breaking those small molecules into their final, breaking those medium-sized molecules into their final small monomers, which we then can absorb. All of which is, or I should say most of which, is completed before we leave the small intestine. Anatomy-wise, the small intestine itself is a very long tube, equaling up to about 20 feet in length. Now, depending on whether those measurements were taken in an individual that's living or an individual that's dead, that length can actually vary. As when you're alive, you have smooth muscle contractions that shorten the length of the small intestine, whereas when you're dead, those muscles are no longer contracting and the small intestine appears much longer. It has a very, very large surface area, as we'll see here shortly, and it's broken in general into 3 regions. The duodenum is the first 10 inches approximately. The jejunum follows for about 8 feet, and the final ileum being about 12 feet. It ends when it joins with the large intestine at the colon, or at the cecum, with the junction occurring at the ileocecal valve. So taking a look at the surface area of the small intestine, we have extensive adaptations that increase the surface area of the small intestine from the microscopic scale with our microvilli on individual cells. Those microvilli the cells then form large villi, which we see here as this finger-like brush covering, and then those microvilli are further covering circular folds in the small intestine, all of which increase the surface area and allow for greater diffusion and absorption. Looking at the histology of the small intestine, the majority of the cells are going to be absorptive cells with microvilli. Those microvilli serve to increase surface area, and they also create what's called a brush border. If we look at the surface of our cell, the lumen I should say, so we're going to have our simple cuboidal cells with our microvilli on one side, and then we have our simple cuboidal cells on the other side with our microvilli here. Okay. Well, sorry about that, folks. My pen stopped working. So again, here we have our surface cell with our microvilli. These microvilli not only increase the surface area for absorption, but they also create this region just on top of the microvilli called your brush border. This brush border is a thin layer of fluid that is trapped in and amongst the microvilli. This brush border is where all of the final, and my pen stopped working again, okay, where all of the final levels of absorption take place. So all of the final levels of absorption, digestion take place within this brush border, which makes sense because once we have once we have broken it up into its last smallest piece, it's then time to absorb it. So we're going to want to do that absorption right here next to the absorptive cell. See if, well, that doesn't do it. Okay. There we go. Well, at least now I know I can fix it without restarting the program. Okay, sorry about that, folks. So, again, let me just go back and finish up here now that I've got my pen fixed. So this small liquid layer on top of the brush border is where the final digestive act takes place. And this is immediately followed by absorption. Okay. Now, the majority of the cells lining the small intestine are these absorptive cells with microvilli that also secrete digestive enzymes that are going to finish that final level of digestion and follow it with absorption. Intermingled among the absorptive cells, you're going to find goblet cells. Goblet cells are going to secrete mucus to aid in lubrication and to maintain the integrity of that brush border. Also distributed amongst the absorptive cells are your enteroendocrine cells. These again are going to be those that secrete the hormones associated with the small intestine. These include, thus far, the ones we've talked about, include secretin, cholecystokinin, and gastric inhibitory peptide, all of which are secreted for different reasons, which we've already mentioned. Finally, you'll also find some panther cells, which secrete lysozymes and have antimicrobial functions. Now, looking at the overall functionality of the small intestine, again, we're going to see two aspects to digestion here. So let me redraw my little absorptive cells here. So here we have our absorptive cells. Here is your brush border. And then in this area you have the lumen. Now the lumen is where digestion is occurring. With the enzymes and secretions from the liver and pancreas. With that in mind, as the fluid in the lumen gets digested from big to medium, it slowly incorporates or gets switched out with your brush border region, where those enzymes within the brush border finish up that digestion, going from medium to small. In many cases, if we take and kind of expand our brush border enzyme here, so there's our brush border, there's our microvilli. In many cases, these brush border enzymes are actually anchored in to the microvilli. Okay, this is so, and I kind of like to draw it as a little scissors, okay, as our little enzyme here. This is so we can take our molecule, break it up, and immediately absorb it into the cell, which with the anchoring of the enzyme into the cell membrane of our brush border cell, this kind of couples that final stage of digestion with the immediate absorption, okay, of the molecule. Now, again, some digestion does occur in this lumen area, this open gap, this open space, but that digestion that occurs there is due to the enzymes secreted by the pancreas and the liver that were dumped in at the duodenum. So let's take a review of everything that's been digested as of yet, starting first with carbohydrates. Carbohydrates, again, can be anything from starches and other strings of large molecules to individual sugars. We start with amylases, which are coming from the mouth and from the pancreas. And these guys are going to break the large starch molecules into medium size. So maltose, maltulose are polysaccharides. Not as large as starch, but still not an individual sugar molecule. Okay. Now what we're going to see is these small or big to medium molecules are then brought into the small intestine, where our brush border enzymes, which you notice, maltase, okay, alpha-dextrinase, all of these guys, are going to break these maltose, alpha-dextrins, etc., into the individual monomer that we are then able to absorb in the form of glucose, or in some cases, galactose and fructose, which are isomers of glucose. Okay. Lactose intolerance is a very common disorder associated with the inability to absorb the sugars found in milk, so you're going to want to read about this in your book for independent study. Protein digestion begins in the stomach. In the stomach, we first use hydrochloric acid to denature and unfold our proteins. And once our proteins are unfolded, we can attack those peptide bonds between amino acids, first with pepsin. Once we move into the small intestine, we have pancreatic enzymes that further break up those peptide bonds. Those pancreatic enzymes include trypsin, chymotrypsin, elastase, and carboxypeptidase. But again, these are going to break our large proteins into smaller polypeptides. The final break of those polypeptides into individual amino acids is going to be done by your brush border enzymes in the form of aminopeptidase and dipeptidase. Lipid digestion, again, this guy's kind of weird because we do have an enzyme that's secreted in the mouth, but remember, it doesn't become active until the stomach. So lipid digestion actually begins in the stomach when lingual lipase is activated, as well as gastric lipase from the stomach. This is going to break up those fatty acids to a certain degree, but their digestion is limited since they are lipids, and the majority of your digestive system is going to be water-based, which is where bile comes in. Bile is an important emulsifier for the fats. Emulsification is a process where we break big fat big fat droplets into small fat droplets. Small fat droplets are definitely easier to handle than the big ones that would occur when we start mixing water, the lipids, with the water that is found in our alimentary canal. We find some additional lipases as well coming from the pancreas to aid in absorption or aid in digestion. Absorption is a little different for lipids, as you will see shortly. As such, we have no brush border enzymes because we don't actually absorb lipids in the same manner that we absorb all of our other nutrients. Lipids are absorbed into the lymphatic system, as you recall, in a slightly different manner. As such, we don't have any brush border enzymes involved since they're not going to be absorbed like all of the other nutrients. Finally, nucleic acid digestion actually begins in the small intestine with enzymes from the pancreas. We have ribonuclease and deoxyribonuclease that first start to break up those large DNA and RNA molecules. Then in our brush border, we have further enzymes that break up those nucleotides into their three components, breaking off the sugar, the phosphate, and the nucleotide nitrogenous base. Those are then absorbed along with all of our other nutrients and incorporated and sent out into our body to be reused and recycled. Now, this table here gives you a nice overview of all of those digestive enzymes we just spent all that time talking about. You are going to want to be familiar with where the enzyme comes from, its name, of course, and what it targets. Great thing about most of these is they tell you that in the name. Lingual lipase, for example, comes from saliva, targets lipid. Okay, pancreatic amylase comes from the pancreas, and for those of you who have been paying attention, anything with an amylase in it, AMYL, targets starches. Now, you are going to want to be familiar with all of these enzymes, and you do want to have an idea as to whether the enzyme is big to medium or medium to small as far as the products. Okay. In general, everything above the small intestine, so everything in this area here, for the most part, is big to medium, whereas down here, it's medium to small. bringing us to the other half of the story. And at this point in my semester, I usually end up running out of time, and as a result, the concept of absorption gets assigned as independent study. Now, why am I assigning this as independent study? Well, in all honesty, we've already talked quite a bit about absorption. We just didn't talk about it as absorption, we talked about it as reabsorption, right? In the urinary system. So we talked about the absorption of all of these molecules when we talked about their reabsorption in the small intestine. And notice we're using the exact same mechanisms here. Secondary active transport, typically involving sodium or hydrogen ions, where possible. And remember, from the urinary system, when it comes to absorbing something, we typically are going to be active at some point, whether it's getting it into the cell or getting it out of the cell. We have to be active. We have to employ energy. Note, for the majority of the nutrients, they're going to be absorbed via into the blood and sent off to the liver. The lipids, of course, absorbed into the lymphatic system. So you're going to read through this section on your own and be familiar with the general absorptive mechanisms for the major nutrients. I did go ahead and leave a lot of the information from my previous lectures in here for you to guide your studies so that hopefully you're not quite as lost. Here we see some nice diagrams showing how protein digestion occurs at the different regions. And here we see another diagram for lipid digestion and absorption, which hopefully is helpful. Okay, so here we have some questions, which I'm going to go ahead and go through here. But if you haven't reviewed digestion, you might actually want to pause the lecture here and come back to these questions once you've had a chance to read over the section on absorption, so these questions actually make sense. But I did actually talk about this one, so we can, of course, kind of review this one. All nutrients get absorbed into the blood capillaries within the intestinal villi. Is this true or false? Well, hopefully you are thinking that this is false. This is, of course, because your lipids get brought into the lymphatic system. So that's, of course, what makes this false. Now, this one you may have to look up, since you probably haven't, if you have not taken my advice and paused the recording. But coming back to this, all amino acids, dipeptides, and tripeptides are absorbed via primary or secondary active transport. Well, if you go back to the diagram from a few previous slides, you will see that this is a true statement. So this one is should be, you shouldn't have to do a lot of reviewing for this one, because the answer to this is actually the same as it was in chapter 26 of the urinary system. So let's see what you all can remember. So, glucose absorption from the lumen of the small intestine occurs via simple diffusion, primary active transport, 2-sodium glucose symporters, or 3-chloride glucose antiporters. Well, it is the same as what we saw in chapter 26, which is 2-sodium glucose symporters. Okay, we are nearing the end of this lecture and of the digestive system, bringing us to the large intestine. The large intestine begins at the ileum with the ileocecal valve and extends up towards the liver with the ascending colon, transverses the abdominal cavity and the transverse colon, and then falls with the descending colon and finally sigmoid colon before it meets up with the rectum and anus. So technically the rectum is the last 8 inches of the of the large intestine, extending finally to the anus or anal canal, which is the last 1 inch of the alimentary canal. Anal canal and the anus itself are surrounded by 2 sphincters. The internal sphincter is an extension of the muscularis of the large intestine, which serves an involuntary control for defecation. The external sphincter is an extension of the muscles that form the pelvic floor and is of skeletal muscle, which of course gives you conscious control over defecation, which we'll talk about that mechanism here shortly. Mechanical digestion in the large intestine is primarily going to be, again, that mixing, squeezing action associated with the muscular action of the large intestine. This muscular action not only is going to compact and compress the fecal matter as it extends through the large intestine, it's also going to aid in the absorption of water and electrolytes as it's squeezed out of the fecal matter as it's being propelled through the large intestine. We have more localized haustral churning that is going to churn up and help absorb the nutrient, the electrolytes and water. And then varying degrees of peristalsis, which is going to move the fecal matter through the alimentary canal or through the colon, depending on what's happening in the rest of the digestive system. There are actually 2 reflexes that involve the colon as it pertains to what's happening in the stomach. You have the gastroileal reflex and the gastrocolic reflexes. Both of these are triggered when the stomach is filling. The idea with the gastroileal reflex is that when the stomach gets triggered or filled, it triggers the emptying of the ileum. Vice versa, that same stomach filling triggers the emptying of the colon. Both of these reflexes surround the idea that we need to make room for the food that's about to come through. So again, filling of the stomach causes emptying of the ileum into the colon, and then anything in the colon into the rectum for expulsion. This, of course, is why many individuals, after eating a large meal, about an hour later typically have to go to the bathroom. This because of that gastrocolic reflex. This information or this knowledge can also be quite useful when bathroom training children, as particularly if you kind of train them to go to the bathroom in the morning right after breakfast, as that first meal of the day can trigger the movement of all of that fecal matter from the previous day into the colon, and you can get your bowel movement cycled like that. There is no human-caused chemical digestion that occurs within this large intestine. The large intestine uses enzymes. If any chemical digestion, I should say, occurs within the large intestine, it occurs due to enzymes secreted by bacteria. This bacterial fermentation not only produces those obnoxious odors that we sometimes have to deal with, but it also produces beneficial side products such as vitamin K and vitamin B. As far as absorption within the large intestine, really there's very little nutrient absorption that occurs here. What is absorbed is going to be any vitamins, etc., that were produced within the large intestine, and then the main absorption occurring here is going to be water and electrolytes. This occurs as the fecal matter is compacted, and the water and electrolytes that were secreted earlier are reabsorbed. So the primary electrolytes absorbed here include sodium and chloride, and of course anytime we absorb sodium in particular, but any of these electrolytes, water will follow. Fecal formation typically takes anywhere from 3 to 10 hours depending on how mobile your colon is, at which point 90% of the water has been removed from that chyme and put back into the blood. As the feces moves through the colon, it is transformed from the liquid chyme to semi-solid by the time it hits the transverse colon, to a more solid mass as we enter into the descending and sigmoid colon. Feces itself is not just the food we could not digest, but it's also quite a bit of other things that you may not expect, including the dead epithelial cells from the lining of your large intestine or from your entire digestive tract, any undigested foods, and then finally, bacteria, whether live or dead, are found in fecal matter. Now, although the large intestine does not necessarily have a large job in absorption, it is significant enough to be important in water reabsorption, water absorption, I should say, where if you suffer from repeated bouts of diarrhea, you will have issues with dehydration. So the large intestine can be quite important in that. Bringing us to the physiology of the large intestine, which is defecation. Defecation is the process of which we expel that unneeded fecal matter from the body. So the elimination of the fecal matter from the rectum is referred to as defecation. Like urination, defecation and the reflex for defecation is centered within the sacral region of your spinal cord. And again, there are a lot of similarities between your urination reflex, micturition reflex, and defecation. It starts with the activation of stretch receptors within the rectum. Sorry, I gotta get my pen back up. Stretch receptors within the rectum detecting the presence of fecal matter. Those stretch receptors signal the sacral region of the spinal cord where the defecation reflex is activated. Parasympathetic signals from that sacral region are going to come down and inhibit the smooth muscles within the internal anal sphincter, and hopefully you are about to find a bathroom where you can also voluntarily relax that external anal sphincter for defecation. Of course, that may not always be possible. This is why we have that external anal sphincter for control over defecation. Although it is not something you want to control too heavily, since defecation problems are quite common. Diarrhea is often only associated with any infections associated with the large intestine, whether it be food poisoning or some type of flu. But diarrhea occurs when the food moves too quickly through the digestive system and there isn't adequate time for the absorption of water. Constipation, though, is much more common, which occurs when the fecal matter stays too long in the alimentary canal in the large intestine, at which point too much water has been reabsorbed, and as a result the fecal matter becomes very hard and difficult to move. Constipation typically is easily handled in two ways. First and foremost, water. If too much water gets reabsorbed, well, then you need to consume more water, because there's a reason why that water is getting sucked out of the fecal matter, because you're not consuming enough and your body's trying to balance it out. You can also increase your water content with the help of fiber. Fiber acts to draw water into your fecal matter, but unless you actually consume additional water along with that fiber, it is very useless, because fiber can actually constipate you if you are not also consuming water. So if you suffer from bouts of constipation, the first thing you need to do is increase water intake. If regular increases in water intake don't help, then you need to add fiber. But you cannot add fiber without water, because it will actually has a potential to make the problem worse. So our final topic here is, how do we regulate this whole process of digestion? Well, we regulate this whole process from start to finish in 3 phases of digestion. And these include the cephalic phase, the gastric phase, and the intestinal phase. Here we have a nice diagram showing everything that's going on in each phase, but let's talk about each individually. Starting first with the cephalic phase. The cephalic phase is the stomach is getting ready. This is initiated by sensory receptors in the head, whether it be sights, smells, or sounds of food. All can be used to activate the cephalic phase. Of course, taste also comes into play as the food hits the stomach, or food hits the mouth, excuse me. The cephalic phase is used to not only prepare your mouth for consumption, but also to get the stomach started out. So the stomach is going to start making noise, getting revved up, increasing juice secretions. Again, any of your special senses can activate this cephalic phase. Nerves involved typically include your facial and glossopharyngeal, which is going to get those salivary glands revved up. Of course, the vagus nerve is going to head on down into the stomach to activate it. Once we actually start consuming food, we then are in the gastric phase, which is the stomach is working. The gastric phase begins when food actually enters the stomach. It is, under normal circumstances, it's regulated independently via the enteric nervous system, primarily, secondarily via hormones coming from the stomach in the form of gastrin. So the primary activation of the stomach occurs for two things. We have stretch receptors, which detect the presence of food in the stomach as it stretches to hold the food. We also have chemoreceptors within the stomach, which detect the increase in pH as the food that you're consuming and the liquids that you're consuming are diluting the gastric juices, causing the pH to rise. The stomach working begins with increased secretions and mixing waves that become more and more vigorous until we squirt a little bit of that chyme into the duodenum. At which point, as you learned earlier, the stomach will actually shut down for a little while as the duodenum deals with the chyme that it just received. In addition to the enteric nervous system, you also have endocrine influences on the stomach in the form of gastrin. Gastrin is secreted again due to distension and an increase in pH, and it stimulates increased gastric secretions and increased intestinal motility. Gastrin will stimulate stomach activity, while at the same time, hormones from the duodenum will inhibit stomach activity. What we basically end up seeing is the gastric phase along with the intestinal phase actually end up playing tag team. So what ends up happening is when the gastric phase is revved up, this intestinal phase is pretty empty, waiting for it to receive something from the stomach. Vice versa, when the stomach phase is revved up, the gastric phase is on pause, since we can't keep shoving stuff into the duodenum when there's no room. So the intestinal phase, as the name implies, begins when food enters the small intestine. And again, a variety of receptors here are used to activate it, both mechanoreceptors that detect stretch, as well as chemoreceptors that detect the content, the chemical content of the chyme that is entering the duodenum. We see hormonal influences here include gastric inhibitory peptide, cholecystokinin, and secretin will all inhibit stomach activity while the duodenum deals with the secretions that it has. And again, we saw the reflexes from earlier. The gastrocolic and gastroileal reflexes from earlier help the intestinal phase function as we attempt to make room for secretions coming from here. Now, like we had gastrin influencing the stomach, we have secretin, cholecystokinin, and gastric inhibitory peptide influencing coming from the duodenum and influencing it. So let's go ahead and go through all of our major hormonal influences, starting with gastrin. Gastrin is secreted from the stomach in response to an increase in stomach pH and or stomach stretching, both of which are signals to say, I just ate something. Okay. Gastrin then enters the bloodstream and targets the gastric glands and the smooth muscles of the stomach to increase the mixing waves and secretions of juices into the stomach. Secretin is released from the duodenum in response to a drop in duodenal pH. This drop in this low pH within the duodenum stimulates the release of secretin, which targets your pancreas to secrete pancreatic juices rich in bicarbonate, as well as stimulating the liver to secrete bile rich in bicarbonate. Lastly, it has an inhibitory effect on the stomach, causing a decrease in the secretion of gastric juices from the stomach. Cholecystokinin, also released from the duodenum when chyme rich in fats and proteins enters the duodenum. CCK targets the pancreas to stimulate juices rich in enzymes. It increases the flow of bile from the liver to stimulate or to be used for lipid digestion. And finally, it inhibits or decreases stomach emptying, inhibits stomach activity so that the duodenum can deal with the chyme that it already has. Finally, gastric inhibitory peptide is also released from the small intestine due to high fat and, in particular, high sugary chyme. This is going to, as the name implies, inhibit gastric activity to ensure that the stomach can deal with the duodenum can deal with what it currently has without receiving an extra batch, so to speak. It also tells the pancreas to release insulin. This insulin, of course, will soon be needed for the use or the maintenance of the blood sugar that's about to all be dumped into the blood from that high sugar meal. So here we also see another diagram that will hopefully give you a nice overview of the different phases as to what is going on, as well as their hormonal influences. So hopefully, depending on which of these diagrams you find most useful, one will help you understand and learn these phases. And another one. I always like pictures because some individuals will find pictures more beneficial and others like the word or written or verbal description. So hopefully I'm giving you each of you something that will help you, if not one way, another, help you learn the material. So a few questions here before we finish up this lecture. Blank stimulates the pancreas to release bicarbonate-rich juices. Is it gastrin, secretin, cholecystokinin, or gastric inhibitory peptide? Well, if you recall, only one of these was secreted due to a drop in pH from the duodenum, which would of course make sense that we would want bicarbonate from the pancreas, and that is secretin. Blank is released due to chyme rich in lipids. It stimulates contraction of the gallbladder while inhibiting gastric emptying. So which one of these was released because of fats and had a fatty function? Is it gastrin, secretin, cholecystokinin, or gastric inhibitory peptide? Well, we've kind of eliminated secretin from the mix because that was the answer to the previous one, which means it's going to be either gastrin, cholecystokinin, or GIP, and the only one that makes sense is cholecystokinin. The gastrocolic reflex initiates mass peristalsis, intensifies peristalsis in the ileum, forces chyme into the cecum, is initiated by secretion of gastrin, or occurs segmentally in the haustral. Well, based on the name, gastrocolic reflex. This is a reflex between the gastric, stomach, and the colon. Of these, the only one that has anything to do with the colon is the initiation of mass peristalsis. The idea here is we are emptying the colon of its food in order to make room for what's coming make room for the new meal we just consumed. As you smell food, your mouth begins to water and your stomach rumbles. This signals the beginning of the gastric phase of digestion. Well, if you're smelling food, does that have anything to do with the stomach? Nope, this would be a false statement because it would be the cephalic phase. Another hormone question. You know I love those hormones. Take a second, read through this, and see what you come up with. So gastrin stimulates secretions of large amounts of gastric juices, strengthens contraction of the lower esophageal sphincter. Gastrin secretion is stimulated by a low pH and is inhibited by a high pH. Well, of all of these, the only one that is correct is D. 3 is not correct. We would have to reverse these. Is stimulated by a high pH and inhibited by a low pH. Remember, we want the stomach to have a low pH. So if it already has a low pH, we don't need any more. If we have a high pH, we're going to need more more enzymes, more hydrochloric acid. So we would need to stimulate secretions at high, but inhibit at low. Okay, that was the last question, and this is the end of your recorded lectures for your AMP2 career. I enjoyed recording these for you. Hopefully you found them useful. If you, again, have found any errors or have any issues with these recordings, please, by all means, email me so I can correct them or rectify them. Other than that, good luck studying for your last couple of exams and your finals.